Okay, well, thank you very much. John and Elizabeth brought me out to dinner, and we had a wonderful dinner, and we spent the whole dinner talking about horseshoe crabs. So I don't have anything more to say about horseshoe crabs. So, but I want to thank you very much for that. Well, first of all, let me just read one short passage in my book, Crab Wars. And this is chapter 3, and it's called First Lessons. And it takes place at the Boston Children's Hospital in 1956. And it goes like this. Mrs. Sargent, your son is all right, but I'm afraid there's been some brain damage. He's talking some nonsense about worms, crabs, and bugs. Oh, that's all right. All he ever does is talk about worms, crabs, and bugs. So just to give you a little introduction to me, I wanted to read that passage. Now, first of all, I'd like to ask, how many people here think, or does anybody think their life has been saved by a horseshoe crab? Okay, nobody? Okay, one person. How many people would have taken a flu shot last year if they were available? Okay, a few more people. How many people ended up taking a flu shot, or have ever had a flu shot, or have ever gone to the hospital and had something come in contact with their blood system? Okay, everybody who raised their hands because they would have taken the flu shot, your lives were saved by horseshoe crabs. And everybody who has had a flu shot, your lives were protected by horseshoe crabs. It's actually about the most common procedure in medicine today is based on the blood of horseshoe crabs, and it's a test for gram-negative bacteria. And it's the only commonly used medical procedure that's based on a single species of wild animal, and that animal is disappearing up and down the East Coast. So we're really running into a fairly critical situation. So now what I'd like to do is take you down to our study site. First of all, does everybody know what this is? Cape Cod? No, that's my left hand. Now, if we could have the lights off. And I will take you down to Pleasant Bay. And Pleasant Bay lies right on the elbow of Cape Cod between Orleans and Chatham. Okay, and this is Pleasant Bay. A lot of these early photographs were taken back in 1979 when we were working on a NOVA film about a year in the life of Pleasant Bay. Now, the interesting thing about Pleasant Bay is that 90% of it is less than 6 feet deep. So it's sort of a boater's nemesis, but it's a naturalist's delight. And this also makes it one of the best places on the East Coast for catching horseshoe crabs because the water is very clear and it's very shallow. So all you have to do is just reach out of your boat and grab them or wade along and pull them up and put them in the boat. This, of course, is Nauset Beach, which is the barrier beach that protects Pleasant Bay. And, of course, that was whacked in our recent northeasters. Now, what we're going to do, Channel 5 lent us their helicopter to go up and film what I think what is really one of the most fascinating scenes in natural history that you can see in this area. And we're going up over Nauset Beach again. This is where a winter storm has pushed some of this sand out into the bay. And we're going to go to this little parabola of sand that has some marsh grass protecting this. This will also show you how shallow Pleasant Bay is. Meanwhile, offshore, a female horseshoe crab has just pulled up out of the sediments, and she's starting to make her way towards the shore. They do this around the full moon and the new moon tides in April, May, June, and a little bit in July. You can see as she's moving along, she's surrounded with these little manidia, the little silver sides that are feeding on the food that she stirs up as she's moving along. As she gets closer to shore, she's joined by a smaller male crab, and they're starting to make their way up to the beach. And again, this is right around the full moon and the new moon high tides. When she gets up right at on the beach, she will start digging down into the sand and placing her eggs in the sand, and she'll be surrounded with about a dozen male crabs that are all jostling around trying to mate with her. And now one of the interesting things about horseshoe crabs from a scientific point of view is their eyes. And I understand we have been talking about that here with Bob Barlow. Basically, horseshoe crabs have about 10 eyes. And they have these two compound eyes here, and then they have a series of other eyes. And most of what we know about human vision and a lot about the human nervous system comes from research on the optic nerve of the horseshoe crabs that runs from these compound eyes to the brain, and the brain lies in a circle around their stomachs. So these guys actually eat through their brains. I don't know if any of you do that. I did it tonight, and I do it much too often. But there was a Dr. H. Keffer Hartline who won the Nobel Prize in 1967 for his research on the optic nerve of the horseshoe crab. And again, this is the large compound eye, and it has about 100 perfectly formed little lenses are part of those two compound eyes. Now, one of the things that's very difficult when you're trying to film horseshoe crabs is they consider anything in the water to be a large female horseshoe crab. So as you're trying to hold the camera steady, each foot will be surrounded with about 30 or 40 lascivious male crabs that are all trying to mate with your toes and everything. So what we would do is one person would be in charge of filming, and then two people would be in charge of keeping them off your feet. But anyway, we were able to get some shots of them. And then as the tide started to go out, what happened is you had about 300 or 400 panicking horseshoe crabs that got caught in this parabola of sand. They all started to swim in a clockwise direction, which is kind of interesting, and then somebody could do a PhD thesis on why they happened to swim in a clockwise direction. A few would then make it out over the ridge and back out into the bay, again the larger female in front and the smaller male behind. And the ones that didn't make it out would then dig down into the sand and wait until you got the next high tide of the following high tide. And if you were to go back into the geological record, not only would you find tracks exactly like this, but you would find horseshoe crabs exactly like the present horseshoe crabs. So they're often called living fossils, and they go back about 350 million years. So they were here way before the dinosaurs, before birds, before fish, before mammals, before almost all the animals that we're familiar with. And basically they used to come up on land, and the only thing on land were little ferns and mosses and a few insects, things like dragonflies with about a 4-foot wingspan. But again, and it's because of this ancient heritage that they're of such interest to scientists. Then the eggs stay in the sand until you get the next full moon high tide a month later, and then they will hatch out as what are called little trilobite larvae, which gives you another idea of their ancient heritage. They are related to trilobites, and of course trilobites are the first animals that developed eyes. You can see here the compound eyes developing and a couple of the simple eyes up there. They even have an eye right out on the end of the tail. Now, the other thing that's of interest about horseshoe crabs is their blood. If we have a wound like this, we have a whole system of antibodies that go to the area and fight the infection. What horseshoe crabs have are a single kind of what are called amoebocyte cells that simply go to the area and coagulate and keep the infection out. So it's a very primitive system, but it's very elegant, and it's worked for 350 million years. So it works particularly well. And basically what happened in the early 70s, scientists learned that you could extract the blood of the horseshoe crabs, separate out the amoebocyte cells, lyse them, and make what's called limulus amoebocyte lysate, which is used as a test for gram-negative bacteria. And the bottom line of all of that is that a quart of horseshoe crab blood is worth about $15,000. So they're basically the most valuable marine animal that we have in this area. And when the industry first got started, we had some funding from Earthwatch expedition, and we went out and we would simply catch the crabs, hold them in these large pens. They would be bled, and then they'd be returned to us, and we could monitor their recovery. And this is the bleeding operation here. This is Stanley Watson, who started the first company called the Associates of Cape Cod. And you can see these are the female horseshoe crabs being held in this wooden rack up here. And then you have a needle that's about the size that a vet might use on a horse, and you just have a free flow of this blood, and this is the blue copper-based blood that is coming out of them. And again, as John was saying, theoretically, you should be able to go out, bleed the crabs, give them a little chicken soup, and they'll, recover their amoebocyte cells and then release them back out, and everything will be fine. Unfortunately, under industrial conditions, quite often the trucks won't show up, they'll be left in the sun, all kinds of things like that, and you'll have multiple traumas. And we were finding sometimes you'd get as many as 50% of the horseshoe crabs would die from all of this. And this just simply shows you that horseshoe crabs like to mate with anything. So that was in the early years, back in 1979. Now I just want to give you a quick overview of the different ways that horseshoe crabs are used. Back in the 1930s, in Delaware Bay, they were used for the fertilizer industry, and they would take out hundreds of thousands of horseshoe crabs and then crush them up and use them for fertilizer. You'd like to think that they stopped doing this for environmental reasons. Actually, it was because they made such a horrible stench that nobody could stand living on the shore. So they finally shut down the fertilizer industry. And the other thing that they're used for is for bait, as John mentioned. And it's kind of interesting. Part of the reason for this is when we stopped, we fished down the conch populations in the Bahamas and the Caribbean, and conch is used in sushi. And the way you go out and catch conch around here is you use horseshoe crabs for bait, and they simply catch the horseshoe crabs and chop them in two. You can see this is horseshoe crab, two halves of a horseshoe crab there, and they use them for bait. So what you're having there is a situation where you're getting the seafood industry, the sushi industry, which, as everybody knows, is a big and growing industry, coming in conflict with the biotech industry with lysate. And of course, this is the lysate industry, and that's the little gel that forms when you bleed out the horseshoe crab, and it comes in contact with gram-negative bacteria. And it forms a gel like that, and it has that very distinctive blue color. This is just simply shows you sort of the evolutionary story for the reason that they developed the amoebocytes is they have little white flatworms called Delura candida, and quite often if you look at a large horseshoe crab, you'll see these flatworms crawling along the shell. And what they do is they lay their eggs in the tissues, the gills of their book gills, and then when they hatch out, they can tear the tissue, and then you get the water coming in, and shallow warm waters like that we have around here that the horseshoe crabs are in are absolutely full of gram-negative bacteria. Gram-negative bacteria include things like E. coli, so they're one of the most common bacteria that we have. We have E. coli in our guts, and it doesn't bother us as long as it stays in our intestine. But if you have a trauma, if you get into an accident or something like that, and your system starts to break down, and those E. coli can get out of your intestines and into your bloodstream, that's when you go into septic shock, and that obviously can be fatal. So that's one of the, and when I talk about a gram-positive and gram-negative diseases, that you split all your bacteria into gram-positive and gram-negative. There was a Dr. Graham who came up with a dye test, and it basically splits them. Okay, so what happened right around the year 2000 is the Cape Cod National Seashore decided to ban the taking of horseshoe crabs on the eastern side of Pleasant Bay. And the interesting thing about Pleasant Bay is they've been catching horseshoe crabs in the bay for the entire history of the lysate industry, over 25 years now, and catching them only for lysate. So it was a very good kind of experimental situation. So both the Cape Cod National Seashore and the Monomoy Wildlife Refuge down on Monomoy put a ban on the catching of horseshoe crabs, and the Associates of Cape Cod brought a federal suit that was deliberated up here at the federal courthouse, the Joseph Moakley courthouse. And when this happened, I really hadn't been thinking about horseshoe crabs for about the last 10 years. And I started to look back at it, and I wanted to kind of figure out what was going on. And I talked to a friend of mine, George Buckley, and we had been working for a number of years together down on Pleasant Bay. And one of the things that he used to do is he would take his Harvard students out, and they would walk along the rack line, and they would simply count the empty shells of the horseshoe crab. Quite often, if you see a horseshoe crab like this on the beach and you think you've found a dead horseshoe crab, if you look carefully, if there's a crack along the leading edge of the shell, that's where the horseshoe crab has crawled out and left that empty shell behind. Actually, my belt buckle was made out of the test of a horseshoe crab. And this just simply shows you when they are molting, and you can see this one down here, and they usually do it down in the sand, and that shell breaks open, and then they crawl out, and he's just left that empty shell behind. And then they swell up by about a fourth. They just take in the water, and then they start to harden, and they'll be about a fourth larger, and then they continue their life. So ones that are about this age, these are probably about 3 years old, will do it maybe once a summer. In their first year, I think they actually they will molt about 14 times. And then as they get older, it's about once a summer, and then as they get older than that, then they finally stop molting. But you see all of these empty shells along the shore, and by simply, counting all the horseshoe crab shells in a 100-meter transect, you can get a pretty good idea of the trends of the horseshoe crabs. And from this, George was saying to me, well, the population of horseshoe crabs has crashed in Pleasant Bay. But I said, George, it's just not true. If you look around, the bay is absolutely full of large horseshoe crabs. We went back and forth, and I said that his field technique was no good, and he said I didn't know what I was talking about. We went back and forth. We couldn't figure out what it was. So then we went back, and again, this is what it looked like back in 1979 when we were working on the Nova film, and you can see a single female surrounded with about a dozen male crabs. Another shot, single female here, single female there, and again maybe 20 male crabs surrounding her. And this is what it looked like in 2001. A single female with a single male and maybe one satellite male that was coming in. And this was the average number that you would see. So something had changed really quite dramatically. You could also see they're not as healthy looking as they had been before. They have a lot more epiphytes that are on them, and the water quality has gone down a little bit. This is the Associates of Cape Cod where they're bled. And this is the new building. I like to say that it's about a year's worth of horseshoe crabs from Pleasant Bay. You can get about $10 million worth of lysate. You get about $250 worth of lysate each time you bleed a crab. So that's about a summer's worth of horseshoe crabs to build their new building down in Falmouth. And actually, when I was working on the book, they weren't particularly pleased to have me, going in and out of the lab. As a matter of fact, they wouldn't talk to me on the telephone, or they wouldn't allow me in the lab. But I did find there was a little hill on a graveyard on the opposite side of the street, and with a very long lens, you could kind of take a photograph right through. And this was the main lab where they did most of the bleeding. And again, this is the old way they used to do it, where they would simply exsanguinate the blood, taking all the blood out of the crab, and then you'd get very high mortality. Now they do that free flow that we talked about. This is Norm Wainwright, who some of you have heard, who was the chief scientist at the Associates of Cape Cod. And this again is the lysate. And then we started figuring out, well, now where are these crabs coming from? At the time, the company was saying, well, that 60% of their crabs came from, and about 60,000 crabs came from Pleasant Bay and Monomoy. And I knew from my past books and from my past experience that they couldn't be in business if they were only using 60,000 crabs. It just didn't make sense. So we started looking into it, and I started trying to figure out, well, where are the crabs coming from? This is the wharf down right off of Woods Hole, and you could see this is a fish company from Fairhaven, Mass, that is picking up some horseshoe crabs that have come out of Vineyard Sound and takes them to the lab. And again, if you collect them, they should be covered with a nice wet piece of burlap. You could see this burlap had come off, and that horseshoe crab is beginning to dry out, and he probably won't make it. So that's one of the, just very simple things that often doesn't happen. And of course, they're loaded into a trash can. So you get a number of crabs. And, a lot of these crabs are probably about 10 pounds each, and they're crushing down on each other, so you can get trauma and little, can break their shells from that. And then we started figuring out where the crabs were coming from. So it turned out there were about 40,000 crabs coming from Monomoy, about 28,000 coming from Pleasant Bay, another about 10,000 crabs were coming from Dennis, so about 60,000 in total from the Cape. But then it turned out, I remembered that back in the 70s, they went from catching all of their crabs in Pleasant Bay to catching the same number. They went from catching 35,000 crabs in Pleasant Bay and another 35,000 from Rhode Island. So then it turns out by 2000 they were getting about 60,000 crabs from Rhode Island and another 10,000 crabs from Chincoteague Bay down in Virginia. And of course, when you're transporting them that amount of distance, the mortality is going to rise as well. And again, so we went back, and this just simply shows you the mortality that we found, again up to about 50% coming back to us. And so then we went out and we started to measure the crabs. And all of a sudden, because I remembered that the crabs seemed to be larger than what I had seen before. So we started to measure the crabs along the carapace. And this is simply the way that you do it. And then I remembered that the fellow called Carl Schuster had measured horseshoe crabs all the way from Maine all the way down to Florida. And he measured both the males and the females across the carapace. And one of the places that he had it for was Pleasant Bay. And so we went back and we started to compare the numbers. And all of a sudden it turned out this was the size of the male crabs, exactly the same as they had been back in the 50s. This was the size of the female crabs, exactly the same that they had been in the 50s. But this extra little hump came from female horseshoe crabs that were simply too large to come from Pleasant Bay. This was the largest crab that you ever found in Pleasant Bay before, and all of a sudden about a fourth of the crabs, probably about 20 or 30,000 crabs, were too large to come from Pleasant Bay. And again, this just simply shows you this was about the average size in the 50s, and that's about the average, or that's the size of the larger ones that we have in there now. So basically, we figured out, well, what was happening is they were being introduced from Monomoy. The same guy who's collecting them collects them both in Monomoy and on Pleasant Bay, and there's a huge difference in size from the two locations. And so we started bringing this evidence to the company, and they said, well, maybe they got in there naturally. Well, if they got in there naturally, what they would have to do is crawl north about 2 miles, and then south about a half a mile, and then north about 4 miles to get up into Pleasant Bay. Theoretically, yes, they could do it, but if the same guy is catching the crabs and bringing them to be bled and then comes back, it makes a lot more sense to release them right in front of his house than to take them all the way back to Monomoy. And then, in fact, we found that all the companies had been doing that. And the reason this was, according to the Food and Drug Administration, when the industry first started, they passed a regulation that said you were supposed to return the crabs to the water from which they came. And so this is the way they would hold them in Pleasant Bay. and they had about 15 of these little carboys that would be floating around the bay, and each one of these could hold several hundred horseshoe crabs, maybe sometimes even up to 1,000 horseshoe crabs. So they would bring them out and bleed them. And then the other thing that was happening is the horseshoe crabs were being caught on the eastern side of Pleasant Bay, where they would come up to lay their eggs. They always would do it along this shore. 90% of the crabs would be laying their eggs up here, and it was easy to catch them because it was nice shallow water. And then what they would do is they would bleed them and then release them back into the deeper part of Pleasant Bay over on the other shore. And this made a certain amount of sense because it would take them about 30 days to get from the deep water back up to the shallow water to lay their eggs. But the problem was you were removing them from the breeding population because they only have about 4 days out of the month when they can breed. Basically 2 days on either side of the new moon and the full moon. So you were simply taking them out of the breeding population. So this was the reason that we were seeing no immature crabs in Pleasant Bay, because there just simply hadn't been any recruitment for probably about 10 years. But no one had really noticed it because other larger crabs had been brought into the bay. So you kept seeing a lot of adult crabs. So this all became part of the court record. And the judge said, well, we have to make sure that this is actually going on, and we're not sure how important the horseshoe crab eggs are to the bird populations. So we went out and we took some photographs like this, and you can see the horseshoe crabs are laying their eggs here, and this little shorebird is feeding on something. We presume it was on horseshoe crabs. We got pictures of the bill coming up, and there'd be horseshoe crab eggs on it. But the judge said, no, that's not good enough. So actually, Mass Audubon had to go out and catch some of these shorebirds and slaughter them, and then slit open their stomach, and sure enough, they found their stomachs were absolutely full of horseshoe crabs. And there were about a half dozen often endangered seabirds, shorebirds, that are dependent on horseshoe crabs for their migration. And the red knots are one of them, and they'll start their migration down on Tierra del Fuego, and then they'll time their migration so they're on the Delaware beaches during the 2 weeks when the horseshoe crabs are laying their eggs. They'll eat about 140 tons of horseshoe crab eggs in 2 weeks, and that gives them the energy to fly back up to the Arctic Circle. And one of the things that they're finding now is that the birds, because the horseshoe crabs have been declining in Delaware Bay for about the past 20 years, that they're actually getting up to the Arctic Circle, and they don't have enough food sometimes to actually get there. Elizabeth was talking tonight about them actually being so weak that they would drop out of the sky. They just couldn't make it, and they'd end up in the ocean before they got to the land. And of course, they would get up there and they wouldn't have food yet, so they couldn't start laying their eggs, so a lot of the eggs weren't viable. And this just simply shows you what the horseshoe crabs used to be like on Delaware Bay. Unlike on Pleasant Bay, where you have hundreds of crabs on the beach, down there you used to have hundreds of thousands of crabs. The whole beach was absolutely littered with horseshoe crabs. You can see these are some of the eggs that are coming up out of the ground, and there'd be hundreds of thousands of birds. These are the red knots. Some of them have little bands. They were actually banded on Tierra del Fuego, and they would pick them up above the Arctic Circle. And again, there'd be hundreds and thousands of shorebirds all over the place. And actually, the guy who did some of this research actually wrote letters to people up and down the coast trying to find out, because he figured there had to be some central place where the birds were getting their food. And he got a letter back from Delaware Bay, and the guy said, "Yeah, we get hundreds of thousands of birds around here in May." And he couldn't believe it. And the guy thought that that was true every place up and down the coast. But it turns out there are very significant areas like that and this whole species. You get 80% of all the red knots in the world are on the Delaware beaches during the 2 weeks when the horseshoe crabs are laying their eggs. So if you had an oil spill, for instance, you could wipe out virtually a whole species in just a couple days. And then this just simply shows you This may be a little just so story, but you can see the horseshoe crabs down here are laying their eggs, and then you get a lot of silversides that are come in and are feeding the eggs, and you can see this guy has just caught a striped bass. So perhaps there's a direct line of causality in that little scenario. And then, so as we started looking into this, it became, it was sort of became a local tempest in a teapot, and I love these little local controversies. And we kept pointing out what was going on. So then everybody decided that they would start doing some studies. As a matter of fact, the reason for this one is the state wanted to, all of a sudden, they were saying, well, our regulations have worked, and that the numbers of catch of horseshoe crabs had gone down from about 400,000 down to about 128,000. And the reason for that is before all the states got together and decided that they would limit the number of horseshoe crabs that would be taken, they asked each state to get a census of how many horseshoe crabs were taken. And of course, what they did is they asked the fishermen, and everybody knew that a cap was going to be coming, so everybody sort of exaggerated, and they doubled and tripled and quadrupled the number that they had actually been capturing. So Massachusetts reported that we had been capturing about 400,000 horseshoe crabs. And then the cap went into effect, and then they started doing research to find out how many crabs were being caught, and they said there were about 128,000. And they said, well, where were the horseshoe crabs? Where were the other horseshoe crabs? And they said, well, the reason for that is that there were so many cranberries that all of the freezers were full, and so people weren't catching any horseshoe crabs. But it was actually just kind of a numbers game. As a matter of fact, now the two worst states that have the worst policies for horseshoe crabs, the sort of rogue states, are Massachusetts and Virginia. Virginia, because they've got this huge bait fishery, and Massachusetts has passed what's called rent-a-crab policy, and that means that the fishermen can go out and catch the crabs, sell them to the lysate guys, and then turn around, get them back, and turn around and sell them to the bait guys. So you're going from what had been, 100%, a potentially 100% sustainable, no mortality fisheries to a guaranteed 100% mortality. And I always make the point that if you cut up the horseshoe crabs and use them dead for bait, they're worth about 30 cents. If you keep the crabs alive and use them for lysate, they're worth, each horseshoe crab is worth about $3,000 over its lifetime for lysate, because you can bleed them every summer and return them back into the water. And so anyway, what happened, a number of studies were done. A group called the Friends of Pleasant Bay, and it turned out the guy who collects the horseshoe crabs was one of the directors of the bay. But anyway, they put up about $65,000 to go out and essentially refute some of the things that we were coming up with. So they did a study of the population of horseshoe crabs. Then the Department of Interior, the Cape Cod National Seashore, spent about $20,000 to go out and study the horseshoe crabs in Pleasant Bay. And then George Buckley and I looked at each other and said, listen, we've got about 25 years worth of data and we've never published it. So we finally took 2 days off and we gave ourselves $200 and we wrote up this third study of the horseshoe crab population in Pleasant Bay. And I like to think that, talk about peer review. We had very top, high-level paid government and private lawyers that were looking at all of these papers. And I like to think that our paper was the one that finally settled the case. And actually what happened on that first study, what they did is they set out a grid and they would simply count all the horseshoe crabs on each point on the grid. And they would count them from June, July, August, right through the summer. And then these are some of the research that the National Seashore did. And this is simply where they would catch the crabs over here and then tag them and then they would get the tags over here. And you can see what the horseshoe crabs do is they spend all summer coming up here to the eastern coast to lay their eggs, and then they go back to the deep water, and then they come back to lay their eggs some more, and then they go back to the deep water. So all summer long they're doing this, and of course what they were doing is they were going right over those points. So people were counting them when they went to the east, and counting them again when they went to the west, and counting them when they went to the east again. So you've got about 4 times as many horseshoe crabs in the bay as in fact I think there actually were. So they basically said that there were 500,000 horseshoe crabs, large adult horseshoe crabs in Pleasant Bay, which would give it more horseshoe crabs than any other estuary up and down the East Coast. And this is a tiny little bitty estuary as opposed to Delaware, where you get the largest crabs and the most crabs in Delaware Bay, and then they get smaller and fewer as you go both north and south. And this just simply shows you, usually in the horseshoe crabs you see one clutch of eggs that is just about ready to be hatched, and then another clutch that is just starting to mature. So there's a possibility that what you have is 2 populations, one population that's laying their eggs on the full moon and another that's laying them on the new moon. So they're coming up and developing the eggs for a month later. And then this simply shows you basically what happened is the federal court case went through, the bans were upheld, and the next year, much to my surprise, all of a sudden we had been seeing virtually no horseshoe crabs all along the rack lines for about the 10 or 15 years, and all of a sudden they were coming in even better than advertised, and you were just seeing hundreds of little immature horseshoe crabs because they were actually having the possibility of mating. So it worked even better than we thought it was going to. And then this just simply shows you the 3 companies that catch the horseshoe crabs: the Associates of Cape Cod, BioWhitaker in Maryland, and Endosafe down in Charleston. And what's happening now is that all of the companies are collecting a lot of their crabs in about a 100-mile area between Cape May, New Jersey, and Chincoteague, Virginia. So what's happening is there's a lot of pressure on these on the horseshoe crabs in that central area. The other thing that happened, of course, was 9/11. Right as I was finishing my book, and all of a sudden I realized that all of the vaccines that we're developing against bioterrorism all will have to be tested with horseshoe crab blood to see if they're contaminated with gram-negative bacteria. And this is a canvas, which simply means, 2 Cambridges. It started in Cambridge, England, and it's now moved to Cambridge, Massachusetts. It actually should be by canvas, but that's a little grammatical quibbling. But they have the contract to make the smallpox vaccine. And again, they'll be testing all of that with the horseshoe crab blood. And then this just simply shows you some new research that's been going on where they're trying to use genetic engineering to replicate that clotting mechanism. And you actually find two biological cascades. One is for factor C, which goes down to form the clot when it's exposed to gram-negative bacteria, and this one over here actually is reactive to molds and fungus. So they're seeing, and I think Norman Wainwright went into that in quite a bit of detail. But the interesting thing about that is, I remember back in the 70s, people were saying, well, this whole controversy will die because somebody somewhere is working on a way of using artificial means to make the lysate. And then in the 80s, it got a little bit more sophisticated, and they said, well, somebody in the Far East is working on a way of synthetically forming limulus lysate. And now they're saying that the guys in Singapore has actually developed the way, and that patent is held now by BioWhitaker. But the problem with that is it takes about $800 million to develop a new drug, to develop something new like that. And from the people that I've talked to, they've said the problem with that is that it's going to be more expensive. You're going to have to use new machinery, and it's not going to be as sensitive as the horseshoe crab test itself. And I don't know if how many people are familiar with the Genzyme building down in Cambridge. It's a big sort of Dickensian building right on the side of Store Drive. And that building, if you look in there, there are these huge copper fermenters, and that building was built to use gene splicing to develop drugs. But then they found what they could do is take those genes and put them into goats, and now they have a little herd of goats out in western Massachusetts, and all you have to do is milk the goats, and your drugs come out in the milk. So you don't need that huge building. So the bottom line of all of that is that if they found a genetic way to make the limulus lysate, what they'd want to do with the gene is then put it in an animal, and it's already in the animal. So we already have an almost perfect system, and if we just deal with it in the right way, have very stringent regulations, and protect the crabs, then I think everything will go on. If you'll excuse this little sort of hippie 60s little horseshoe lobster man here, I just want to make the point that we have other species that are also very lucrative, and we have very, very stringent regulations. I'm sure everybody realizes that basically what we're doing with lobsters is we have size quotas on them. So after they reach a certain size, you can collect them. So basically what happens is we feed them. We put bait down into the crabs, and the lobsters will go in, and they'll eat the bait, and then they come up, and we throw them back, and they eat some more, and they get bigger and bigger. Once they get a millimeter over a certain size, then we can keep them. And actually in some states they have an upper limit. So if they can get by that, then they're safe on the other side. So it's sort of actually it's a form of aquaculture. We're just feeding the lobsters, and as it turns out, as we're catching more lobsters, the natural population seems to be increasing as well. So it's sort of a large aquaculture type situation. And essentially what we have with horseshoe crabs is we have what's called ocean ranching. So what you're doing is the crabs are living in the wild, you're taking them out, you're bleeding them, and then you return them back to the wild. And if you do that properly, everything will be okay. And here we are, the sun is setting, and the horseshoe crabs are mating, and I thank you very much. Okay.
I'll do the lights a little slowly, so are you ready? Watch your eyes.
And I'll turn that off so we can hear.
Thank you very much. Do people have questions? I'm sure there is.
Yes.
Could you clarify again the difference in the population you said before the court made the decision on regulating? Was there a difference in population before and after the decision? I didn't understand.
Yes. Basically, you could see it in those photographs. You'd see there were a lot. The breeding aggregations were way down. But what had been happening is that large crabs were being introduced to the bay, so that you were kind of getting an artificially large number of adult crabs. But because they weren't able to breed, you were getting no replacement crabs. You were getting no recruitment going on. So two things were going on at the same time. Let me just say one thing, and that's because I kind of missed it as we were going along in the slides. The other two companies, Endosafe and BioWhitaker, actually have little what are called raw lysate facilities. And what they do is they go out, they catch the crabs right at the spot, and then they bring them back and they bleed them, and then they release them again. And that's when you get virtually no mortality. Just for historical reasons, the Associates of Cape Cod, because they started, having it all centralized, they do everything right at the company. And so that requires, catching the crabs hundreds of miles away and bringing them up and bleeding them, and then either getting them back or turning around and selling them to fishermen. So that's where you get very high mortality. Yeah? To revive very quickly, you just put them in the water and they, aerate their gills, and they recover very quickly. But it takes about 30 days for them to rejuvenate their amoebocyte cells.
So how does that compare them to that cycle of meeting?
Yeah, I see. It's a very good question. Often I draw the analogy, it's a little bit like taking all the women that are about ready to have babies on Cape Cod, catching them, putting them on the space shuttle, sending them up to the International Space Station, taking 30% of their blood out, and then putting them out in Worcester, and then see how many can get back to the Cape. So that's kind of what we're doing. I thought it was a little digression from what your question was, but no. In fact, you find that they're very disoriented when you bring them back. You put them back into the water. Nobody's done really definitive research, but it certainly looks like even if they could get back on the beaches, they wouldn't be able to lay their eggs. So you're just that amount of trauma. You're getting mortality. Yeah.
Just in that same kind of similar question.
Yeah.
Also about pledging numbers.
Yeah.
Are there certain companies, these companies that pledge that 30% amount of drop of the bleeding? In other words, I'm assuming you're saying that was kind of my question. Is there a regulation on how much?
Basically, the way, and again, it's another sort of very common sense thing. They just they're upside down in their rack, and they do a free flow of blood. And when the blood stops flowing, then they stop. And it turns out that's about 30%. So, which is better than the old way when they took everything out. Yeah. It's not an exact science, is what. Yes. Yeah. They get to be about 20 years, and the reason we know that is they raised some female horseshoe crabs at Wellesley College. You only have women at Wellesley anyway. And what they did is they kept the shells, and they have about 20 years worth of the cast-off shells, and that seems to be the same case in the wild. Just to give you an idea of the difference in sizes, a 20-year-old female horseshoe crab in Delaware Bay will be about this big around and weigh about 20 pounds. On the Cape, it will be about 10 pounds and be about that big. Up in Ipswich, where I am now, it'll be about that big and weigh maybe a measly 5 pounds or something like that. But they'll all be 20 years old. Yeah.
Is there the blood that comes from a horseshoe crab, is there a difference between the female blood and the male blood? Is one more desirable?
Yeah, that's a good question. The reason they primarily bleed the females is simply because the females are larger. The reason they're larger is they keep shedding their shells and they keep growing. The males stop growing when they're about the size of your hand. So it's just simply a matter of time and money. You can get more blood out of a large female than you can get out of the males. Yeah.
Which sex is more common?
In the wild, it appears that everybody assumes that it's probably 50-50. 50% are males and 50% are females, the way it is in most species. But when they're breeding, I think what happens is each time they breed, all the males are on the beach, and you get and half of the females are breeding on the new moon, and half of the females are breeding on the full moon. So that's why you get a lot more males around each male. Whatever. I'm getting really confused. It's genetic. Anyway. Yes.
Are there any other fisheries in the world besides East Coast of the United States?
Oh, for horseshoe crabs. Yes, they do catch them. You do have horseshoe crabs. There are 4 different species of horseshoe crabs in the Far East. Basically, you only get them on the east coast of continents or the west the west side of oceans. The reason for that is that's where you get the warm currents. So we get the Gulf Stream that creates this nice, long, warm niche for them. So you get the horseshoe crabs here. In the Far East, you get the same thing off of Japan, and they have about 4 species over there. They have used them for lysate over there, but it turns out the Lemulus polyphemus that we have is better. Actually, the Associates of Cape Cod has been bought by Segakaku, which is a very large Japanese firm, and they actually made most of their money by using shark cartilage. And probably a lot of you, I use shark cartilage for joints, and nobody knows why it works, but it works. Okay. Yes?
Is there any danger that the horseshoe crab will ultimately become extinct if present trends continue?
I don't think, not biologically extinct, but what is happening is because most of the collecting is done in small areas, you're getting population extinctions. So you're getting, and it turns out that, those are the areas that you want to protect the most. As in fisheries, they're finding the best way, rather than trying to have quotas and all of these things that everybody gets upset with and they don't work very well to protect the species that you want to protect. It seems that a sanctuary area, like we had down here, they also created a sanctuary off of Delaware Bay. That seems to be the best way to protect them. And particularly if you're protecting them so that you can't catch them when they're coming up to lay their eggs, that makes perfect sense. So no, I don't think we're not concerned about a biological extinction. I primarily see this as a medical problem because we are really, a lot of our medical profession is dependent on horseshoe crabs. I mean, it's protecting us. I mean, horseshoe crab, the test is used every day, thousands of times a day, everywhere from third world clinics to major pharmaceutical companies. So it is one of the most common procedures in use today. Yeah. Show us.
How do you know, or how do they know if they've already taken the horseshoe crab? Do they tag them, or how do they
Yeah, you can actually, after they've been bled, you can actually see the puncture wound. And then actually what happens sometimes is a little fungal growth will grow around that wound. And so sometimes that can even prove to be fatal over time. So you can actually, I've done some research on myself just to see, how long you can see after you've had a shot. And you can, in a human, you can see it for about 2 weeks. So you can probably see it for at least that amount of time in a horseshoe crab. Now they do have some regulations so that you're supposed to tag them. When we were doing some of our research, what we would do is we would simply clip. They have the little spines off of their tail, and we designated each spine as a specific week in the summer when they were bled, and then they would clip that spine, so then you would know when they were bled and everything like that. The good thing about that is that it was very, nobody else knew what you were doing. So people didn't know that the crabs were being tagged, and we could start getting this information. But now, after you bleed them, you put a regular tag on them.
How was this tagged?
They have a couple. I know some of them was as simple as just simply taking a rubber band and putting it around the base of the tail. They also have tags that's it's a little one that just goes into the side of the shell. And they were even working on a kind of a brand, sort of a cold, I don't know whether they use liquid nitrogen or something like that you could brand them, extending the ocean ranching analogy, I guess. Yeah.
What is the mortality?
They basically, pretty much everybody was admitting that there was about a 10% mortality. And I think it depends a lot on, again, with the raw lysate, you're going to have a lot lower mortality. You're going to have very high mortality the further you transport them. And if you're doing what Massachusetts is doing, where you turn around and sell them for bait, you're getting 100% mortality. Yeah?
Besides what other predators do horseshoe crabs?
Just about, well, when they're tiny, everything eats them. They're like little ice cream cones or something like that. Birds eat them, crabs, anything that can get their hands on them. Once they get to be about the size of my belt buckle, they're pretty safe. Occasionally, you'll find a very hungry tiger shark will have maybe a couple horseshoe crabs in it. But once that's why they've survived for 350 million years. They've got this nice hard shell, and they're not very appealing. I have been asked by some people, when they're making, cooking cookbooks, for some non-profit organization like that, and they've asked me if I could give them a recipe. So I give them a recipe for horseshoe crab chowder, and you cut up potatoes and onions, and then you add a pound of crow meat, and then they usually figure out that it's maybe not exactly kosher. Okay. Are there any other questions? Nobody's asked me. Yeah.
Do you have copies of your books for sale?
Oh, of course. That's a good. Yes. And how much did you? Huh?
What's the story with your belt?
It was made by a woman in Chatham. I actually bought it in Woods Hole at a place called Under the Sun. You can get them around. I'm sure you could still get them down in Woods Hole. It might be a good thing for the aquarium to have. But
It's a little
yes, the story. Yes, it's basically they just simply took the test, the molt of the horseshoe crab, and used that as a basis for the mold, and then they just poured the liquid metal into it. A little bit more tricky than that, but that's pretty much. I think actually what they did is they put it in wax to make the mold, and then they poured it in there.
One last question. Who wants the last one?
I haven't. Yes.
How big an issue is habitat destruction?
I think a lot of people say there was some research that was done in the Far East, and they were saying habitat destruction was a big issue, and groins and seawalls and things like that. It's certainly something to be concerned about. In an area like Pleasant Bay, you had an 800-pound gorilla. So basically, I think what was happening in Pleasant Bay is that basically about 90% of the horseshoe crabs were being taken out every summer and bled and then returned back into the bay. So I think that was the dominant thing that was going on, and that the habitat, frankly, down there hasn't changed all that much. There are some more houses. There is more nitrogen in the bay, but there are a whole lot more horseshoe crabs in the bay because they were being introduced. So I don't think that was a factor there. It's something to be concerned about, but I think there are much bigger things going on. The bait industry, the lysate industry, that have a much bigger effect. Okay, so nobody asked any hard questions.
Well,
Good.
They're just holding them for practice. Bill, thank you very much.
Okay, thank you.